Despite the importance of Mendel's work, there are several exceptions to most of his principles. For example, not all genes show simple patterns of inheritance. In most organisms, genetics is more complicated because the majority of genes have more than 2 alleles. Also, many important traits are controlled by more than one gene. So as you can see, the key concept for this section is that phenotype is affected or influenced by many different factors. So we're going to talk about incomplete dominance and codominance in this section. As you can see, incomplete dominance is a type of inheritance in which one allele for a specific trait is not completely dominant over the other allele. This results in a combined phenotype, which again is the physical appearance. For example, in the picture you guys see here, snapdragon plants. So if you cross-pollinate a red snapdragon with a white snapdragon, you're going to end up with a pink snapdragon. So a cross between, so here are 4 o'clock plants. And this is a pretty common exception to Mendel's principle. So some alleles are neither dominant nor recessive. So as you can see here in the picture, the F1 generation, which are all these guys produced in here, produced by a cross between a red flower and a white flower, you get pink flowers. Which allele is the dominant allele in this case? The answer is neither. Cases in which one allele is not completely dominant over another, again, is called incomplete dominance. In incomplete dominance, the heterozygous phenotype lies somewhere between the 2 homozygous phenotypes. Most of the time you can remember it as being blended. So if you take white and you blend it with red, you're going to result in pink. So this is incomplete dominance, where there isn't one allele that's dominant, so to speak, over another allele. So here we're going to look at a incomplete dominance genetics problem. In northeast Groton, there is a creature known as a wildcat. It comes in 3 colors, blue, red, and purple. The trait is controlled by a single locus gene with incomplete dominance. A homozygous, or big B, individual is blue. A homozygous recessive, little b, individual is red. And a heterozygous dominant, big B, individual is purple. What would be the genotypes and phenotypes of the offspring if a blue wildcat were crossed with a red one? So what we're going to do here is we're going to actually work it out. So in your notes, what I want you to do is try to work it out yourself. Again, we're looking for a blue wildcat, big B, being crossed with a red wildcat, big B. So I want you to pause the video and work out that square. So we're going to take our big B, and our red guy, which was big B. So we're going to get big B, big B, big B, big B, big B, big B. So as you guys can see here, so since big B equals blue, we're going to have 50% blue. And down here, we're going to have big B, which yields red. And again, we're going to have 50% of there. So the genotypic is a 1-to-1 homozygous dominant to heterozygous dominant. And the phenotype would be blue to red. So here we have another example of incomplete dominance in the color of betta fish being shown. So these are our cute little betta fish. When a green fish, and they call green fish being B1, is crossed with a steel blue fish, B2, all of the offspring have the heterozygous genotype B1, B2, in that blended color. These offspring will be a royal blue color that comes from the phenotype from both alleles. The alleles of this gene follow the pattern of incomplete dominance. So if you were to cross these two, if we did another square, you're going to get 25% to be green, around 50% will be royal blue, and another 25% will be steel blue. Now we're going to look at codominant alleles. First, let me point out the meaning of the prefix co, which means together. Think about it. You have cooperate, which means to work together, coexist, exist together, cohabitate, habitat together. The genetic gist to codominance is pretty much the same as incomplete dominance. A hybrid organism shows a third genotype. Not the usual dominant, and not the quote-unquote recessive one, but a third and different type. With incomplete dominance, I'm going to call that ID, you get a blended. You get a blending of the dominant and a blending of the recessive traits, so that the third phenotype is something in the middle. Like when we went back and looked at the red and the white plants and we got the pink. In codominance, the recessive and dominant trait both appear in the phenotype of a hybrid organism. So you get them both. And you're going to see what I mean by that as we go forth and do some of these examples. So a typical example showing codominance is the ABO blood group system. For instance, a person having the allele A and B allele will have blood type AB because both of the A and the B alleles are codominant with each other. So remember that blood cells carry antigens. And an antigen is a molecule that can trigger an immune reaction on their surface. So human blood type A carriers have A antigen, type B has B. If you have type AB blood, you get both antigen A and antigen B. And as you can see, if you are a type O, you get no antigens for you. Because this trait comes from 3 different alleles, A, B, and O, not only is this a codominant alleles, but it's also known as a multiple allele trait. So the multiple alleles shown in the picture are called IA, IB, or lowercase I. Some people with type IA will have the A antigen, whereas if you just had the lowercase I, you would have type O with no antigens. So many genes have more than 2 alleles. And so traits produced by 2 or more genes are polygenic traits. Poly meaning many, and genic means genes. For example, here you see eye color. Traits produced by 2 or more genes are polygenic traits. Poly meaning many, and genic meaning genes. For example, here you can see our order of eye dominance and eye color. Brown is dominant to green, which is dominant to blue. So obviously, brown is dominant to blue. So these genes do not account for changes in eye color, changes in eye patterns, or continuous range of colors over a lifespan. Therefore, scientists hypothesize that there are still undiscovered genes that somehow influence eye color. But for now, we'll know these 3 genes in the order of dominance being brown, green, and then blue. So, no gene is an island. Very rarely do genes even work alone, and very rarely are traits controlled by one gene, as we mentioned earlier. Think about how many genes it takes to make your face, like your cheekbones or the shape of your lips. Genes interacting with one another makes Mendelian genetics even more complicated. So here we have an epistasis, or an epistatic gene. Epistasis is Greek for standing upon. One gene expression modifies or suppresses another gene, again showing us that no gene is an island. So the first gene is going to do something to another gene. Lots of examples. For example, in this picture, albinism is a single epistatic gene. It interferes with the expression of several other genes. So as you can see in the picture, albinism is characterized by a lack of pigment in the skin and the eyes. This mouse is homozygous for the alleles that prevent the coloration of fur, which is white. Regardless of the phenotype, it would normally come from other 4 genes. A person with alleles with albinism may have very light hair, very light skin, and eyes, regardless of the other genes that he or she inherited. So the key thing to take away from this particular slide is that an epistatic gene can and does interfere with other genes, which can have an effect on the organism as a whole. So phenotype is more than just the sum of gene expressions. One of the best examples when we look at how phenotype is a combination of your genetic makeup and environment. Did you know that the female sea turtle makes a nest on the beach and buries her eggs in the sand? Eggs that mature in warmer temperatures develop into female turtles, and eggs in colder temperatures develop into males. Isn't that interesting? Height is an example of a phenotype strongly influenced by the environment. So you can either be tall or short based on your genes. So identical twins, we tend to believe they're genetically identical. However, genes do not control everything, especially the environment during the early stages of development, which can have long-lasting effects. One twin might get more nutrients, and the other, due to the position of the fetus in the uterus, may not. Differences in nutrition and healthcare may also differ height. So, the concepts in this section don't necessarily make Mendel dumb in any ways. There's simply more information that's been discovered and added to his ingenious work, making this snowball we call science just get a wee bit bigger and bigger and bigger.